Okay, now coming to the second video for the calculation of determination of pKa. Now we have noted down in the tabular column whatever the pH we have measured using the instrument here. 0.0 sodium hydroxide to 7.5 ml of sodium hydroxide we have added. Each addition of after 0.5 ml we have noted down the pH of the solution. Now we have to fill this tabular column for doing the calculation part. So for 0.0 there is no delta pH, no delta V, no delta pH by delta V. Okay, now for 0.5 ml the delta pH is the difference in the pH of previous reading and present reading. Please try to understand, the delta pH is the difference in pH of the present reading minus the previous reading. You have to note down here. And the delta V, the difference of the delta V always 0.5 ml. So the delta V is 0.5 ml all the time because we are adding every time 0.5 ml increments of sodium hydroxide. So after filling this column, finding out the difference in the pH of previous reading and present reading, you note down the values here. And the delta V is always 0.5. And then you calculate delta pH by delta V here. So once if you fill this table ready, now you have to take out two graph sheets to plot the graphs to find out the pKa. So take the first graph. So first graph, you will be taking the graph sheet, y-axis and x-axis. And the first graph you are plotting taking volume of sodium hydroxide on x-axis and delta pH by delta V on y-axis. So you have to take the scale, each unit is 0.5 ml of sodium hydroxide added and each unit on y-axis you have to take 0.4 of delta pH by delta V. I'll send you the scanned copy of this graph. So for these readings, you fill the table, plot the graph. So for plotting the graph, you indicate all these points in the graph and draw a smooth curve, and suddenly there will be a drastic increase in the pH, and after that you will be having two readings and you have to plot a graph like this. And from the peak of the graph, you have to draw a straight line onto the x-axis, and this point where it is touching the x-axis is called as equivalence point. So in titration experiment, you are calling this point as end point, where end point is the point exactly where the indicator is changing the color. You are noting down as end point. But in instrumentation experiments, you will call that end point as equivalence point because exactly at this point the neutralization reaction is getting completed. So this is one graph to find out the equivalence point of the experiment. So taking volume of sodium hydroxide on x-axis and delta pH by delta V on y-axis and indicating all the points and plotting the graph. Okay, now you are plotting the second graph. Second graph, again x-axis, y-axis, taking volume of sodium hydroxide on x-axis and pH. You have to take the pH, whatever you have noted down here, on y-axis. And indicate all the points and you will get an S-shaped graph like this. So in this graph, from the first graph, you will be knowing the equivalence point. Here the equivalence point is 6. So exactly at 6 equivalence point, in the S-shaped graph, you draw a straight line onto the x-axis. Again at 6 is the equivalence point. Half of that equivalence point, so exactly half of 6 is 3. So at 3, you are drawing a straight line onto the graph and drawing one more straight line onto the y-axis. Please follow. Equivalence point is 6. Half of the equivalence point is 3. At 3, you are drawing a straight line onto the graph, drawing one more straight line onto the y-axis. Okay. Now, whatever the pH we are observing at half equivalence point, is the pKa value of the given weak acid. Please, again I am repeating, this is very important Viva question. They will ask you, whatever the pH we are observing at half equivalence point is the pKa. Okay, from these graphs there is no separate calculation part. Directly you have to mention here in the observation column the equivalence point 6, half equivalence point 3.0, the pH at half equivalence point, whatever the pH we are observing at half equivalence point is the, you are noting down. So the pKa value of given weak acid is equal to whatever the pH at half equivalence point is the pKa, which is nothing but your result. So the result is pKa value of given weak acid is the acetic acid we have taken. So it generally for the acetic acid the pKa value will range from 4.5 to 5. So between any range 4.5 to 5 you will get the pKa value. Now you will get a doubt how this pKa is related to pH. According to Henderson's-Hasselbalch equation, if you see in the Viva questions, the question number 7, how are the pH and pKa related? So according to Henderson's-Hasselbalch equation, pH is equal to pKa plus log to the base 10 concentration of salt by concentration of acid. This is the equation. At half equivalence point, what happens is the concentration of salt is equal to concentration of acid. So this becomes 1. So therefore, whatever the pH we are observing at half equivalence point, it becomes the pKa of given weak acid. So please follow this carefully, read the Viva questions, you will understand better, listen to the video, you can understand further, and when you are coming here, if you are having further any doubts, I will clarify to you and we will do the experiment in the lab. Meanwhile, you record all these things, do the calculation part, plot the graphs, find out equivalence point, find out half equivalence point and measure the pH at half equivalence point. That is nothing but the pKa value of the given weak acid. So this is the first experiment in your instrumentation experiments, that is the determination of pKa value of weak acid using pH meter.